Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about chromosome working. Before that, many thanks to my friends, those who have already subscribed my YouTube channel. If not yet, please subscribe now. Your subscriptions are a lot to me. Watch and like my videos and also share them with your friends. Chromosome working is a technique used to identify, isolate and clone a gene or a DNA sequence of interest by sequentially analyzing overlapping DNA fragments. This technique is used when the sequence of the target gene is known but there is no clone of the gene. In this technique, we move along a chromosome from a known starting point, which can be a marker gene or a specific DNA sequence, in order to identify and isolate adjacent unknown sequences until the target is reached. This technique is used for characterizing large regions of chromosomes. Chromosome working is a method of positional cloning used to find, isolate and clone a particular allele in a gene library. This technique was developed by Malcolm Bender, Pierre Spirer and David S. Harkness in the early 1980s. Before chromosome working, several positional cloning tests are performed. After identification of the marker gene on either side of the unmapped sequence, chromosome working can begin from one of the markers. The markers means it is a gene which has already been identified or it is a sequence that is already known. Only after identifying a marker gene which is linked with the target sequence, we can start chromosome working. For example, this is a small chromosome which has several genes such as A, B, C, D, E, F, G and H. Out of these, we want to isolate and clone the gene G. We know its sequence, but we do not know its position on this chromosome. There is no clone of this gene. In order to identify and isolate the gene G, we must have markers on the same chromosome on either side of this gene, on right side or on the left side. For example, this is the marker. Without having a known marker which is linked to the target gene, chromosome working is not possible. After identifying the marker on the same chromosome, we can move along the chromosome by sequentially analyzing overlapping DNA fragments until the target is reached. In this figure, for simplicity, the chromosome working is shown in right direction, but in real life, we would move along the chromosome from a known starting point in both directions until the unmapped region is reached. In this example, this is the unmapped region. By chromosome work, we would map the gene G. For chromosome working, first of all, the genome containing this particular chromosome is isolated and is then subjected to partial digestion by suitable restriction enzymes. As a result of this digestion, we will get a number of DNA fragments of manageable size. Many of these fragments will have overlapping sequences. In the next step, These fragments were introduced into a suitable cloning vector such as plasmid. After inserting the DNA fragment into plasmid, the recombinant plasmid is used to transform E. coli. After transformation, the E. coli is plated on a nutrient agar to form numerous colonies. Each E. coli colony represents one clone which contains a particular type of DNA fragment which has been obtained from the restriction digestion of the genomic sample by suitable different restriction enzymes. So in chromosome working, after partial digestion, these DNA fragments are used to construct a gene library. Chromosome working is used to identify and clone a gene from its known closest marker. This technique is based on nucleic acid hybridization. In our example, B is the marker gene. Chromosome working starts from a known starting point and proceeds in both directions until the target is reached. This is the gene library which has several colonies. First step in chromosome working is to identify the colony which contains the marker gene. For this, the master plate is pressed onto a velvet cloth. By pressing, we are transferring an exact pattern of colonies onto the velvet cloth, which is then pressed onto a secondary plate containing nutrient medium. So by doing this, we are transferring different bacterial colonies onto a secondary nutrient plate. This process is known as replica plating. And this secondary plate, after incubation, will develop an exact pattern of colonies as that of the master plate. For identification of all unknown sequences, this procedure is followed. To identify the colony that contains the marker gene, that is the gene B, these colonies are lysed and then hybridized with a probe that can pair with the fragment B, which is present in one of the colonies. Probes are short oligonucleotide sequences that can best pair with the target DNA. As we know the sequence of the marker gene, we can easily develop the probe. The probe can also be fluorescently labeled, by which it will be easy to identify the particular colony. After hybridization, we found that the probe has been bound to this bacterial colony. So we can conclude that this bacterial colony contains the marker gene, that is gene B. After identification, the marker is isolated. Its restriction map is prepared, after which a small part of it from one end is separated and is used as probe 2 for identification of the adjacent overlapping DNA fragment. For this, probe 2 is subcloned. After which it is used for hybridization. As previously mentioned, use of fluorescently labeled probe easily identify the bacterial colony that contains the target DNA fragment. For identification of the adjacent fragment, here also after replica plating, the secondary plate is incubated and the developed colonies were the lysed. The lysed bacterial colonies, each of which represent one clone, are hybridized with the probe 2. After hybridization, if we notice that probe 2 has been bound to this bacterial colony, we can conclude that this colony contains the DNA fragment which should be positioned next to the marker gene. So in this example, this fragment which is positioned after marker gene should be the gene C. After identification, fragment C is isolated from the bacterial colony. So this is the fragment C. Its restriction map is prepared, and after which a small part of it from one end is used as probe 3, which is used for the identification of the next overlapping DNA fragment. So this small part will be used as probe 3. For this, probe 3 is subclone. Probe 3 is subclone, after which it is used for hybridization. It is used for hybridization. For every DNA fragment, its restriction map is prepared in order to find out the orientation of these fragments on the chromosome, as well as to find the overlapping sites. Because in chromosome walking, we are using two different restriction enzymes, the resulting DNA fragments that are obtained due to partial digestion of the genomic DNA will have overlapping sequences, based on which, by sequentially arranging DNA fragments, we can construct the original sequence of the chromosome. After probe 3 is subcloned, we have to find out the next overlapping DNA fragment. For this, the secondary plate is incubated, after which these colonies are lysed. Each colony represents one clone which contains a particular type of DNA fragment. These lysed colonies were then hybridized with the probe 3. After hybridization, if we found that probe 3 has been bound to this bacterial colony, that means this is the colony which contains the next overlapping DNA fragment, which in our example should be the gene D. So the fragment which is present in this colony should be positioned next to the fragment C. In our example, this fragment should be D. So we can say that chromosome walking is a method of positional cloning. After identification of D, the sequence is isolated, its restriction map is prepared, after which a small part of it from one end is separated and is used as probe 4, probe 4 for identification of the next overlapping DNA fragment. In our example, fragment B will have overlapping with fragment C, and fragment C will have overlapping with fragment D, and D will have overlapping with E. So in this way, by sequential analyzing adjacent overlapping DNA fragments, we can easily construct the original sequence of the chromosome. After identifying the target gene, this particular sequence can be cloned for further study. Applications of chromosome walking. This technique is used for positional cloning. In addition to this, it is used to analyze genetically transmitted disease and to look for different mutations. This technique is used to discover single nucleotide polymorphism of different organisms. And finally, chromosome walking is also useful to identify genetic markers. Disadvantages. Chromosome walking is a process which is slow and time-taking. This is due to the fact that the DNA fragments that are cloned in these techniques are small. Another disadvantage is that chromosome walking becomes difficult when repeated sequences are present throughout the chromosome. In addition to this, chromosome walking is not a viable option when the marker is distant from the target gene. Finally, the presence of unclonable sections throughout the chromosomes make chromosome walking not possible. The difficulties arising out of the presence of unclonable sections has been solved by the development of another technique known as chromosome jumping that skips the unclonable sections. Friends, this is all about chromosome walking. For more updates, subscribe my YouTube channel, Sam Biology Class.